Today we will study young stem of Cucurbita. Cucurbita mexicana is a weak stem plant which climbs with the help of tendrils. Following are the important internal structure of the young stem. Epidermis first. It is the outermost layer, single layer composed of thin wall, compactly arranged tubular parenchymatous cell without intercellular spaces. The outer surface of the cell remains cutinized. Numerous unbranched multicellular hair arises from the epidermis. Stomata may be present. Cells are living and contain vacuolated protoplasm. The epidermis is not circular but is wavy, showing prominent ridges and furrows. Second is the hypodermis. It lies next to the epidermis, consists of 6 to 8 layer collenchymatous patches below the ridges and 1 to 4 layer parenchymatous patches below the furrow region. The parenchyma often contain chloroplasts, hence are photosynthetic in nature, but collenchyma remain more concerned with providing protection and mechanical support. Third is the cortex. It lies just below the hypodermis and consists of 2 to 3 layer thick parenchymatous narrow zone around the central stele. Chloroplasts may be found in these cells which carry out the process of food manufacture. Cortical cells usually store the food material. Endodermis. The innermost layer of the cortex is popularly called endodermis, which is a uniseriate layer composed of barrel-shaped cell containing the starch grain. That is why it is sometimes also referred as starch sheath. Steel. Steel mainly involves pericycle, vascular bundle, pith, and interstellar ground tissue. It is a polyfascicular siphonostellic type. Pericycle consists of few layers of sclerenchymatous cell found on the inner side of the starch sheath and is in the form of a continuous band. Some parenchyma cells are also found between the vascular bundle and the sclerenchymatous band, and these are also considered as a part of pericycle. Vascular bundle. In all the 10 vascular bundles are found in the stem, which are organized into two rows. Vascular bundle of outer rows are smaller and occur below the ridges region, while those of the inner rows are larger and placed in the furrow region. Vascular bundle are conjoined by an open type having end duct xylem. Sequence of tissues in vascular bundle in the outer phloem, outer cambium, xylem, inner cambium and inner phloem. The outer phloem is relatively better developed and consists of sieve tube, companion cell and phloem parenchyma. Sieve tubes are well developed and shows perforated sieve plate here and there. The outer phloem is somewhat convex and the inner phloem has semilunar shape. The phloem parenchyma are small and thin wall. In xylem, metaxylem vessels are of quite large size, which shows characteristics of a climber. They have pitted thickening. Protoxylem vessels are narrower and are found toward the center. They have usually spiral and annular thickening. The trachea and fiber are either few or lacking, but xylem parenchyma are abundant. Vessels are not found radially arranged. Cambium occur on either side of the xylem and are accordingly called outer and inner cambium. The outer cambium is many layered while the inner consists of only few rows of cell. In young stem, pith lies in the center and is composed of thin wall parenchymatous cell. In mature stem, the cells of the pith are broken and disorganized, leaving a hollow pith cavity in the center. Medullary rays are parenchymatous in nature. The vascular bundle of the outer rows represent the leaf trace and those of the inner rows corresponds to the true stem bundle, that is the colline bundle. These vascular bundle develop from the procambial tissue of the apical meristem. Secondary growth in climbing cucurbit is restricted to individual vascular bundle and secondary vascular tissue are hardly distinguishable. In such case, outer phloem toward the pericycle consists of primary phloem and toward the cambium the secondary phloem. The inner phloem remain entirely made up of primary element. The secondary xylem lie just below the outer cambium and have much large vessel as compared to primary xylem. Continuous band of secondary wood and secondary phloem is not found in cucurbites. Thank you.